Good morning, everyone. Today I want to share a story of survival, evolution, and the remarkable power of genetics. A story that unfolds on a tiny island in the Galapagos archipelago and centers on one of the most iconic examples of evolution in action, Darwin's finches. But before we dive into the details, let's make sure we understand a few key concepts that are central to the story. Genes, alleles, natural selection, and patterns of selection. Every living organism has genes, segments of DNA that contain instructions for building the body and regulating its functions. These genes influence everything from eye color to body size to beak shape in birds. Different versions of a gene are called alleles. For example, one allele might code for a small beak, while another codes for a large one. An organism typically inherits one allele from each parent, forming a genotype, which helps determine the phenotype, the observable trait like beak size. Natural selection is the process by which individuals with traits better suited to their environment survive and reproduce more successfully than others. Over time, those advantaged traits become more common in the population, and within natural selection, we observe patterns of selection. Stabilizing selection favors intermediate traits. Directional selection favors one extreme over the other. Disruptive selection favors both extremes and selects against the middle. Now that we have that foundation, let's look at how all of this played out in real life on a small island in the Galapagos with two species of finch. Let's zoom in on two finch species living on Daphne Major, the medium ground finch, Geospiza fortis, and the large ground finch, Geospiza magnorostris. In 2004 to 2005, these birds endured a severe drought. Food became scarce and competition for heartsease intensified. The larger ground finches with their powerful beaks had the upper hand. Medium ground finches that had evolved larger beaks, perhaps once advantageous, were now at a disadvantage. Why? Because they were too weak to compete with the larger species and too inefficient to process smaller seeds. As a result, many died, but those with smaller beaks survived. To understand this shift, we need to consider the competitive regimen, what the environment looked like before, during, and after the drought, and how selection pressures changed. Before the drought, G. magnorostris was rare. G. fortis had access to a variety of seeds, and finches with a wide range of beak sizes could survive. This was a time of stabilizing selection, where medium-sized beaks were slightly favored. During the drought, G. magnorostris became more common. They excelled at cracking large seeds and outcompeted large-beak G. fortis. Meanwhile, the large-beak G. fortis couldn't efficiently switch to smaller seeds either. This led to a strong directional selection for small-beaked phenotypes, which had higher survival rates. After the drought, G. magnorostris numbers declined, reducing competition. The selection pressure relaxed, but the small-beaked individuals, the survivors of that intense selection event, now made up a large portion of the G. fortis population. This entire scenario is a real-world example of ecological character displacement, where traits evolved to reduce competition between similar species. But here's where it gets even more fascinating. Scientists wanted to know what genetic factors were behind this survival pattern. Why did some birds thrive while others didn't? Using genomic analysis, researchers discovered something extraordinary. They identified a region on the genome, roughly 525 kilobases in size, that showed strong genetic differentiation among large, medium, and small finch species. A locus is a specific location on a chromosome where a gene is found, and a kilobase is a unit of measurement in genetics that means 1,000 base pairs of DNA. At the center of this region is a gene called HMGA2. HMGA2 is known in other animals, including humans, for influencing body size. For example, in mice, a mutation in this gene leads to a pygmy phenotype, and in humans, variations in HMGA2 affect height and craniofacial dimensions. In Darwin's finches, researchers found that this locus had a major impact on beak size, not beak shape. That's controlled by another gene called ALX1, but the overall size of the beak, which directly affected survival during the drought. When scientists looked at the genotypes of median ground finches that lived on or died in 2004 to 2005, the pattern was clear. Birds with 2 copies of the S allele associated with small beaks had the highest survival rates. Birds with 2 copies of the L allele associated with large beaks were much more likely to die. Heterozygotes, those with 1 of each allele, had intermediate survival. This pattern suggests that the HMGA2 locus had an addictive genetic effect on survival. This describes how different alleles at a specific locus, HMGA2, contribute to variation in beak size and ultimately survival. For example, if you inherit 1 small beak allele, your beak gets smaller. If you inherit 2, it gets even smaller. In fact, a single SNP, just 1 base pair difference near HMGA2, was found to explain 27% of the variation in beak size among medium ground finches. That's an enormous effect for 1 genetic variant, and it gets better. This same locus explained 30% of the evolutionary shift in beak size that occurred during the drought. So what does this tell us? First, it shows that a single gene, HMGA2, can have outsized influence in an evolutionary context. This gene became a genetic focal point during a moment of intense environmental stress, driving a measurable shift in the population. Second, this story reminds us that evolution doesn't just work slowly over millennia. It can act rapidly within a few seasons when selective pressure is strong and genetic variation is already present. And third, this study gives us a clear picture of how genetic architecture can shape the path of adaptive radiation. Adaptive radiation is the rapid evolution of multiple species from a common ancestor, each adapted to a different ecological niche. Darwin's finches famously diversified into multiple species adapted to different ecological niches. The fact that HMGA2 variants existed across species and were shaped by both selection and introgressive hybridization, gene flow between species, suggests that these finches have a shared genetic toolkit that evolution reshuffles as needed. Interesting, the S allele at HMGA2 appears to have come from the small ground finch, a different species that occasionally interbreeds with a medium ground finch. This kind of gene flow, called adaptive introgression, may have played a key role in helping the population respond to the drought. It's a striking example of how evolution can be a collaborative process across species boundaries. Now I want to end with a broader reflection. This research doesn't just deepen our understanding of Darwin's finches. It challenges a long-standing assumption that evolutionary change is always driven by many genes with small effects. Here we see that sometimes a single gene, when aligned with the right ecological conditions, can shape the fate of a population. In this case, a drought on a tiny island turned a genetic variant into a survival ticket. That's evolution in action. And it's not just about beaks. It's about how life adapts, endures, and transforms in the face of change. Thank you.